Welcome, dear viewers, to the video on freeze thaw study for pharmaceutical formulations, pharmaceutical drug products. From long time, I was trying to make the video on freeze thaw stability study. It is also known as a cyclic stress study. So today, I am trying to give you the complete information in the single video. If I get other understanding and knowledge, then I will be making the another video. So be with me till the end of this video, and I assure you that you will get complete information about this freeze thaw stability study. This is somewhat different type of study, and this study is a part of stability study. It is not a formal type of stability study, and it is done for some different reasons. The formulations are different, the reasons are different, the way of doing this study is very different. So entirely it is a different type of stability study done on the pharmaceutical formulations, and this study will give the complete information about the temperature variations. Freeze thaw stability study or freeze thaw stability study is done for assessing the stability of the formulation under temperature variations. As I mentioned, it is a type of study for studying the effect of variation in the temperature. It is also called as cyclic study, cyclic stress study, and freeze thaw stability study. So starting from the definition, a freeze thaw study evaluates drug product resilience or stability to temperature variations. And these temperature variations are not uh, very small. These are the drastic temperature variations, like the freezing temperature and the higher temperature compared to the storage temperature. It is important and essential for liquid products, some sterile products, and some temperature sensitive products, so that we can ensure stability during the storage period and the transport period. Now you might be thinking about the stability studies, which are formal stability studies. So understand the thing that formal stability studies are performed onto the recommended storage temperature, like long-term temperature we call it, then the intermediate temperature and the accelerated temperature. But in the freeze thaw study, the temperature variations are different, or extreme temperature variations are there, and the same product is exposed to the lower temperature and the higher temperature. So this is different study than the formal stability study. Now coming to the definition of freeze and thaw. So freeze means transition from liquid to solid, or it means the temperature variation which lead to the change of liquid to solid, that is freezing, thereby altering the physical structure. Opposite of the freeze is thaw. So thaw means the temperature variation which leads to return to liquid state from the solid state, and potentially it affects solubility and homogeneity. So freezing is lower temperature and thawing means higher temperature. What is the purpose? Why we conduct the freeze thaw studies? Why the requirement is there for the freeze thaw studies? This study is performed to evaluate chemical stability of the formulation. It assesses the physical stability. It also ensures microbial stability, and it also ensures the container closure system integrity at the temperature variations. So first is chemical stability to identify degradation and the effect on potency and the safety. So chemical stability is assessed by freeze thaw stability studies. Then this study is also done to assess the physical stability. It monitors the crystallization, phase separation, sedimentation, and protein aggregation. So for the liquid formulations, if liquid formulation is a suspension or emulsion or a solution, and if we freeze it, there may be chances of drug crystallization. This study is done to ensure the microbial stability, to verify the stability for the sensitive formulations. This study is also done to check the container closure integrity, as there may be the change in integrity of the container and closure system due to variation in the temperature. This study assesses packaging durability through the cycles. It assesses the container stability, whether there is any leak in the container, whether the container gets shrink, or due to higher temperature the container get opened or it loses the integrity. Physical stability considerations involve the crystallization, sedimentation, and denaturation or aggregation in the protein formulations. So crystallization may be there in the solutions upon freezing, and these insoluble crystal may not get back into the soluble form upon thawing. Sedimentation, suspension, emulsion may show separation, phase separation, change in the sedimentation rate, redispersibility, which impact the uniformity. Protein formulations have the risk of denaturation or aggregation during the freeze thaw cycles. Now coming to the another important aspect why the freeze thaw stability studies are done and what is the relevance to the product shelf life or product recommended storage temperature. You may think that if the label instruction is there regarding the storage temperature, then why freeze thaw stability study is to be performed? So the answer to this question is real life scenarios. See, many products are exported from one country to another. So shipping and transport may be there, and there may be long transport time, and during this transport period there may be variation in the temperature. Exposure of the formulation or product to extreme temperature during the transport may happen. There might be intercontinental transport, international transport, and the product may get transferred from one climatic zone country to another climatic zone country. Sometime there may be long shipping time, there may be change in the environment, sudden change in the environment, there may be loss or there may be disruption into the temperature or storage temperature or transport temperature while shipping the formulation. Then in many of the places, daily temperature fluctuations are there. Variation in the warehouse temperature causing the unintended freeze thaw cycles. Sometimes in some regions, day temperature is very different compared to the nighttime temperature. So if you store the product at room temperature, so daytime temperature will be different and nighttime temperature will be different. There may be too cold or there may be high temperature during the daytime. Now coming to the patient product handling and storage. Variability during the patient use such as moving from refrigeration or refrigerator to room temperature. Unknowingly patient may store the formulation in freezer. Similarly, healthcare provider may store the formulation in freezer unknowingly or due to mishandling. Some formulations are required to be labeled as do not freeze or store in refrigerator. So these instructions or labeling instructions are there and those are required to be tested. Applications. So sterile products, injectable solutions, eye drops, vaccines. Then for the temperature sensitive formulations like emulsions, creams, gels, biopharmaceutical formulations are required to be tested for Freeze Thaw Stability Study. The biopharmaceuticals products like protein based drugs and biologics. Nasal sprays are there, eye drops might be there, injectable suspension solutions might be there. Also reconstituted formulations might be there. All these type of formulations should be tested for the effect of freeze thaw stability study. If nasal spray is exposed to the extreme temperature, then uniformity of the drug in the nasal spray may get hampered. So to ensure the uniformity after storage or after temperature change, the freeze thaw stability studies to be done. What are the benefits of freeze thaw studies? So it helps and it provides the quality assurance, regulatory compliance, and optimize storage recommendation. Quality assurance, it identifies stability issues to reduce product recalls. Regulatory compliance necessary for submissions to international markets. Optimize storage, it provides guidance on storage and handling to maintain the quality. After the study, you can provide the labeling instructions very clearly so that you can protect the formulation and protect the public health also. Objective of the freeze thaw studies is to have confidence onto the product behavior upon the temperature changes. So how to perform the freeze thaw studies? Well, first we have to make the protocol. Purpose is assess stability of the drug products under repeated freeze thaw cycles. So this is the objective of protocol or purpose of protocol. And goal is to determine the physical and chemical stability after freeze thaw cycles or cyclic temperature variations or temperature variations. Now what are the requirements? First you need the product samples. Then you need the freeze thaw chamber or you can use freezer and a refrigerator and the accelerated temperature or higher temperature chamber. Freezer with temperatures like -20 or -80 or 228. Refrigerator 28 degree or room temperature and the chamber with higher temperature like 40 degree or 50 degree. Analytical equipments as per the product requirement, HPLC, UV visible, pH meter or dissolution apparatus, and data loggers for temperature monitoring. But for the chambers, I recommend that freeze thaw chambers should be used because these have uniform temperature cycles and these are maintaining the temperature. Then procedure overview. Sample preparation, prepare and record the initial characteristics. Freeze thaw cycle, repeated freezing and thawing. Sampling and analysis, visual and analytical evaluation will be there and data will be recorded and compared. Step 1 is the sample preparation. Prepare samples in vials or final containers. Apply the cycles as per the product requirement. Freeze thaw chambers. Record the initial characteristics like assay, viscosity, pH, appearance, and the other product specific characteristics. See, there is no specific guidance onto the freeze thaw cycles, how many cycles to be performed and what is the temperature. So, as per the product requirement, these freeze thaw cycles to be done. So, freezing and thawing is done and then these cycles are repeated. Freezing involves keeping the samples at minus 20 degree, minus 25 degree or minus 80 degree or 2 to 8 degree for 24 to 48 hours or more based on to the product. Freezing may be done as a slow freezing or rapid freezing. Thawing means thaw at room temperature or slowly thaw at different temperature like 20 degree, 25 degree, 40 degree or 50 degree. Continue the thawing for 24 hours or 48 hours or more depending on to the formulation requirement and your study requirement. Thawing may also be slow or rapid. Means once you have freezed the sample, keep it at room temperature for some time and then expose to the higher temperature. Repeat these cycles for 1 to 3 times, 3 to 5 times or 6 times. So number of cycles are required to be reported. Step 3, sampling and analysis. So visual analysis will be there, visual description, precipitation checking, physical change like color change. So inspect the samples, analyze the potency using validated methods, check the assay and then confirm the data. So physical stability, no significant changes, appearance, phase separation. Chemical stability, assay remains within the predetermined specifications like 90 to 110% or 95 to 105% of the initial content. And impurity levels, no significant increase in the impurities. Then data recording, record all observations systematically, compare results with initial values and prepare a trend or report any trend or deviations. Then conclusion, what is the outcome of the study to be determined if the product maintains quality, safety and efficacy under freeze thaw conditions. And why it is important? Because it is critical for products exposed to variable conditions during transport and storage. See, number of cycles and conditions may vary based on to the product requirement and some regulatory requirements, but no clear-cut regulatory requirement is given. So based on to the product understanding, product requirement and the storage, recommended storage, the freeze thaw stability studies are performed. So this is the information regarding freeze thaw study. Mainly liquid formulations are to be tested for cyclic stress study and freeze thaw study. I hope you might have got good idea about this topic. Thank you for watching the video.